Hello and welcome to Nikolai's Genetics Lessons, and today's problem I got from one of my subscribers, and here's a problem. The following statements are given towards explaining the consequences at the end of meiosis. As usual, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So first of all, let me use couple other drawings to explain what's happening here. First of all, one step before what we see here, usually at the middle of the meiosis we may find chromosomes which looks like this. So this is two sister chromatids, and here we have another pair of sister chromatids. So we call this tetrasome. Basically, we have here four chromosomes that are connected together in the middle, which we call centromere, at the centromere. So basically, what these two sister chromatids represent, this is identical chromosomes. One of these chromosomes is result of copying another chromosome. So this is two identical chromosomes which at this state we call them sister chromatids. And this is homologous chromosome. For example, this one you got from mother side and this one you got from the father side. And also during meiosis it's going to make a copy of itself. And at this state, as you see, now we have two homologous chromosomes. Small changes we can find here, though it is same chromosome, say chromosome number 1, but because inherited from different parents, these chromosomes can be slightly different in some points, may have point mutations, or even deletions, duplications, and so on. And during meiosis, two out of these four, two non-sister chromatids, may exchange their genetic material during process which we call crossing over. Crossing over may happen in many different places, both on short arm and long arm, if chromosome has different size arms. And two of the sister chromatids doesn't participate in crossing over. So the end result would be one parental chromosome intact, one another parent chromosome intact, and also we may get one chromosome that is a result of recombination, and another chromosome also would be result of the recombination, result of the crossing over. So basically now let's analyze what we see on this picture. First let's write down the sequence of one of the chromosome here, and the sequence would be A, so A, then B, C, D, and here this is centromere here, so let's put a dot here to define the centromere. Then we have E, F, G, H, and I. And sequence of the other chromosome or order of genes would be, once again we start from here, so it's going to be A, B, then G, F, E, centromere, T, C, H, I. So it is obvious that these two chromosomes or chromatids are from the different parents. So as you see, this is different because sequence or order of genes is different. So we know that this is not sister chromatids but two homologous chromatids, and cross here represent crossing over. And we also see that on the one of the chromosomes we see reverse order of genes, so from here to here. So basically order have to be A, B, C, D, but instead we see A, B, and then this piece of the chromosome, basically small piece would be on one arm and another piece of the chromosome would be on the other arm. So there was a breakage in this chromosome in two places, and then the central part will just flip over, and then mechanism that repair broken DNA just connected in the wrong way. Will this cause problem? In most cases not, if this breakage wouldn't happen in the middle of the gene, so in this case the gene would be interrupted, but if it would happen in the intergenic space, so it's not going to affect genes, especially also if some regulatory sequences that can lie outside of the gene also wouldn't be affected. So we know that one of the chromosome with normal order this person got from one of the parent, and this chromosome he got from the other parent. And basically this person has two sets of the chromosomes and genes, so this person doesn't have any problem, health problem, but as you see a little bit later I will explain, this may cause serious health issues in the following generation, or most likely it also would cause such serious problem that such child, if we talk about human genetic disorder, wouldn't be able to develop, so would be not viable, and probably this would lead to self-abortion or miscarriage. And basically chances are 50 to 50. As you see, 50% of the gametes would get normal chromosome. Another 50% also would get normal chromosome, even with reverse order of genes. But those chromosomes that would participate in crossing over, and if crossing over would happen within this inverse order of genes region, this may cause serious problems. So take a look, here's another picture. So imagine that this is one chromosome. On purpose I'm drawing with one arm longer than the other one, so you would better understand what happens here. And imagine that this is another one, and once again the order of genes is following: A, B, C, D, then here we have centromere, then E, F, G, H, I, J, and K, And once again, here is centromere of the other chromatid, and order of genes would be A, B, then G, F, E, D, C, H, I, J, and K. So imagine now that crossing over happens anywhere within this region. So let it be here, and in this case results of crossing over would be as follows. So let's start here. So we have A, B gene, C, then we go on other side and we have sequence D, E, F, G, and B, A. So this is going to be a sequence of one chromosome, and here would be a sequence of genes of the other chromosome. So K, J, I, H, G, C, then crossing over, and D. So here we have also centromere E, F, G, H, I, J, and K, and another centromere also present here. So as you see, both sequences, this one that resulted after crossing over has centromere, and another one also has centromere. And basically what we have now, we have one short arm that basically present in two copies. So one chromosome would represent two short arms, but another chromosome after separation would represent a chromosome with two long arms. So once again, let me return to our picture and let's take a look. Again, what would be result of the crossing over? So one chromosome would be A, B, C crossing over D, centromere E, F, G, B, and A, and another one would be I, H, C, D crossing over, and D, centromere E, F, G, and H, I. And of course, also as a result, we would see duplication of basically most of the genes on the, like in this example, long arm, and duplication of genes on the short arm. So basically we are going to see the picture that is not going to be something like this. This would we would see if crossing over would happen normally without when it happens in the region where there is a reverse order of genes, and also this, as you see, region has centromere. This is also important, but actually we would see as a result of such a crossing over one chromosome with two short arms and another chromosome with two long arms. So if we stretch them, we are going to get one chromosome with two short arms and another chromosome with two long arms. And somewhere around centromere we would see this region that is shown here. So basically what you have to memorize that if crossing over would happen within any of these places within this region where reverse order of gene have happened, and if this region also includes centromere, result would be two chromosomes, one with two short arms, another with two long arms. And do not forget that those we have here for chromosomes, gamete would get only one of each chromosome. So we are going to get 4 gametes. Each gamete would have only one chromosome. So this chromosome is normal, this chromosome is normal, but here gamete would get chromosome, say chromosome number 1 with only 2 short arms, and also would get another gamete, chromosome number 1 with 2 long arms. So as you see, we have here duplication, and we have whole arm that is missing in both these variants out of 4. So probably a person who has such a problem, 50% of all the cases when this person would produce gametes or this couple would conceive a child, this would result in self-abortion or stillbirth. So now let's return to our problem and let's try to answer the questions. So variant A, the resultant 2 chromosomes will have deletions and duplications, and this is correct statement. So this is true. So we would have duplications of the same arms, and we would see at the same case that such chromosome would miss one arm. Variant B, decentric and acentric chromosome will be formed. This would be true if crossing over, not crossing over, but actually if centromere would be outside of this region. So if this centromere would happen outside of this region and crossing over would happen within such region, then in this case we would see that one chromosome would have 2 centromeres and another chromosome wouldn't have any centromere at all. So now variant C, the inversion does not allow crossing over to occur, so even if crossing over is initiated, it will fail to occur. Actually, as you see, there is no problem for crossing over to occur here because sequence of 2 chromosomes would be the same due to this structure here. So gene C, gene D, E, F, and G would line up, and nothing would prevent for crossing over to happen. So next, variant D, crossing over is considered suppressed by inversion, as the eccentric chromosome will not segregate normally. So once again, this would be true if centromere would be outside this structure. And variant E, all the gametes formed will cross over. Chromatids at the end of the meiosis will non-viable as they carry large deletion or duplication, and this is true. As I said, probably such an organism wouldn't be viable because its whether would have this chromosome that would have 2 short arms or two long arms. So would have duplications, and at the same time the whole arm, almost the whole arm, would be missed. So this is a correct statement. And variant F, the fragments having non-crossover parental chromatid will survive. And this is also true statement. As you remember, I show you here that 50% of the chromosomes would be intact, would be parental genotype, and 50% as a result of crossing over during tetrasome state would result in such chromosomes with two short arms or two long arms, with duplications and with missing a lot of genes. So this is also correct statement. And now we can choose the correct answer, and as you see, the correct answer would be answer 4. A, E, and F are correct statements. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.